And welcome back to the lab. My name is Rich. Welcome back to Speaking of Diamonds. And speaking of diamonds, today is the much anticipated second part to our scientific experiment on what is on our drills. Now, if you haven't watched part one, I'll go ahead and link it uh, in the description here. And I suggest you probably go look at video one first. But if you choose not to do that, you just want to see this, I'm going to cover some of the facts that we talked about in the first video, as well as some disclaimers, etc. So the reason I'm doing this is, first of all, it is for fun. I'm not a scientist. I am not a germaphobe. I was just interested in when people tell me, and a lot of people have told me, that they lick their pens when they place ABs because the ABs always wants to stick to the wax or the putty that's being used. Now, people do say that you could use older wax, older putty, you can get it, dirty on your sleeve or what have you, and it will be less sticky. One of the ways that I have found a foolproof way of not having your drills stick is to take your pen and blot it on a moist paper towel that I put in a tray, much like this one. I put a little square of paper towel. I wet it, just moistened, and I dab my pen, pick up my AB, place my AB, dab my pen, pick up my AB, place my AB, and it works perfectly. It works for single placing and multi-placing. Well, a lot of people say, yeah, the water thing works, but I just lick my pen. And for the life of me, I just couldn't imagine that was a good thing. Now, what I'm going to show you here today is the results of an experiment in which I took random drills from several different various vendors, and I poured a little bit of each one, including an AB, a fairy dust, and then I think there were two regular packs of regular drills in there. And I took those and I put them in a beaker. I put in a little distilled filtered water, mixed it around, and then swabbed each one of them. And then I put the swab on a petri dish. I then took the petri dishes and I put them in my oven just because that's the warmest place in my house with the pilot light going. And I let them sit there for 6 days because even though that's probably the warmest place in my house, it's winter. My house is like 68 degrees. So my oven is not an incubator whatsoever. So they say that the cultures can grow anywhere from 2 to 7 days. So this took about 6 days. Now, I will say for the first 4 days, there was nothing growing on the petri dishes. Day 4, something started happening. And then it just sort of increased over the next couple days. So here we are. I have them hidden under this cloth because I want to build the suspense. And speaking of building suspense, let's talk a little bit about bacteria, because bacteria is probably the most common thing that we would find on our drills. Did you know that bacteria have been considered the first living organism on Earth? And they have been on Earth for at least 3 billion years. So if anything, we're doing a little Jurassic Park here and finding some of our friends. And we can say hi if we indeed based on what we find here. Did you know the number of bacteria in your mouth is more than the total number of humans on Earth? That's crazy. The length of a bacteria ranges from 0.5 to 5 micrometers. A micrometer, just for the sake of comparison, The human hair, these are all facts that I've looked up, so I'm not making anything up here. The human hair is about 50 micrometers wide. So this is zero point 0.5, or bacteria is zero point 0.5 to 5 micrometers. Bacteria also has the smallest eyes in nature, but in terms of their body size, they are the largest eyes of any type of um, uh, bacteria or organism, I guess I should say. There is not a single bacteria in the body of newborn babies. I thought that was sort of interesting. Um, Babies are born with no bacteria, and then we just pepper them with bacteria as they get older. Now, as I said in my first video, I'm not a germaphobe, and that's truly my belief, is that you have to expose yourself to certain things in order to become resistant to them. Okay. So much like a child, a baby who comes into the world, they're going to get sick. We all know that babies are going to get sick. They need to build up their immunity to not get sick in the future, or to not get as sick as they possibly could. This also speaks to vaccinations, whatnot. That's all very controversial. I don't want to get into that. But essentially, keep in mind, a vaccine is you're introducing the germ or the bacteria into your body, and then prompting your body to fight it off so that it's able to recognize the item the next time it enters your body. So now that I've built up some suspense, are you at the edge of your chair? Let's go ahead, and I'm going to show you what I found based on our swab of. Now, I swabbed the drills twice, the same batch of drills, did it twice just so I could get, accurate reading. And then I did a pen. I did both the multi-placer side of a pen and the single tip of the pen. I swabbed the putty that I had been using for some time, that I was using that pen, and it's been set aside for a while. So let's go ahead and see what happened here. These, my folks, these are not drills. We're going to talk about what these are in a minute, but let me explain to you what you're seeing here. This is so fascinating and exciting. It worked. I'm like really sort of geeking out here. Okay, so first of all, this is our pen sample. And as you can see, and there really isn't anything for me to hold this up to the camera for, nothing grew here that I can see. Okay, there could be something microscopic on here. It could be that it needs another number of days, but nothing grew on the pen. However, our drills seem to have some funkiness going on. And let me bring these up. You got a little bit of a reflection there. Let me try and get it. You can see the camera, whatnot. But basically, they look like little drops of little bubbles of caramel. They're tan in color. And these are both our drill samples, drill 1 and drill 2. Now, let me first tell you, before we go any further into this, these stink. These absolutely stink. When I was first looking at them, one of them came open a little bit, so I've taped them shut. But even now, they're not airtight, and I can smell them, and they smell disgusting. I don't know how else to say this, but they smell disgusting. So what are these? What is this funk growing in here? So I turned to my friend Melanie, Diamonds and Dogs. She is an ex-biology teacher. So not only does she know a lot, but she has all of the good sites and stuff like that she knows of. And she sent me some pictures and some description. And I want to go ahead and bring those up and talk a little bit about what these potentially may be. Now, one thing I want to say is I don't know for sure. Melanie doesn't know for sure. We're taking a hypothesis, an educated guess here a little bit. I also have an educated guess as to why nothing grew here, and it doesn't have anything to do with the fact that your pen just miraculously cleans itself. Okay. Um, but first, let's understand in a lab, what they would do is they would take these samples, they would stain them with some type of dye, and then they would be able to look at them under a microscope and do a positive identification. In this instance, I don't have a microscope. I don't have a dye uh, to put it, to color it, but you clearly can see that there's something here, and it's the same thing. It's really the same thing in um, both petri dishes here. And when I turn them over, it actually looks like little blisters, little tan blisters. So let's talk about so how do I said I don't know what they are. The staining would tell us, and Melanie confirmed this, and I did look it up. The only thing that the staining would be able to tell us, which is really all I'd be able to do here without microscopes and stuff like that, is to tell us whether these are bacteria. They're definitely bacteria. They would tell us if they're gram positive or gram negative. And basically, the difference is that one has an outer membrane, so a negative uh, gram negative bacteria has an additional outer membrane beyond the outside. There's a word for it, but there's an outer membrane. Gram positive does not have an outer membrane. So what the difference is that when you stain them, they would turn different colors because one is going to more readily accept the dye than the other. But what this also tells us is gram negative bacteria are usually more resistant to antibiotics than gram positive bacteria because they have an extra layer of protection. Now, I will also say that Melanie, once she sent me this information, she said, please be careful. This is real stuff. Dispose of this correctly. So, may be freaking out just a little bit here at I don't know what's growing in my petri dishes, hence the gloves and whatnot. But this is the real deal. Okay. So, what these may be is, and I'm going to share, and I'm pulling it up on my tablet over here. What these may be based on the screenshots that Melanie sent me from the different photos on what is this, researchgate.net, that was able to give her some of this information. What she says that this most likely is, and is confirmed by this researchgate.net, is this could be E. coli. And I will pop the pictures up here. You can see that the pictures look very much like the petri dishes we have here. They are a cluster of blister looking like items. And in the picture, the incubation period was 24 hours. And again, they put it in a warm incubator to keep it warm and promote the growth. I had to wait a couple extra days because I don't have that incubator. I didn't want to screw around with putting them on top of uh, a space heater or a heating pad or anything like that and potentially, cook them and make it not turn out. So let's talk about, I know a lot of us, we know what E. coli is, but in case you don't know, E. coli is gram negative. So what that means is they have that additional outer membrane. So the bacteria is more resistant to outside forces, if you will. Gram negative rod-shaped bacterium that's found in the lower intestine of warm-blooded organisms. Most E. coli strains are part of the normal biome of the gut. we all take, or know someone who takes, depending on what your health requirements are or restrictions, take a prebiotic or probiotic, right? So the probiotic is the good bacteria that you're taking to help your digestive system, and it lives in the stomach and it makes everybody happy. What they say is normally after you take a round of antibiotics, they do suggest taking a round of probiotics uh, to encourage the growth of this bacteria again, because the antibiotic will kill the bacteria in your stomach, which is also why sometimes you get a very upset stomach when you're taking antibiotics. So let's see. So it's normally part of the gut. The bacteria are mostly harmless or even beneficial to humans. However, what we know is that some bacteria is very um, is harmful to humans. So we know that we can get sick from E. coli. So while it may be beneficial, if it ends up that you hit the wrong strain, it gets in the wrong place, what have you, different things can happen to us humans. So, how do we prevent getting sick from E. coli? You always wash your hands thoroughly before handling food or after using the bathroom, because we talked about how it's in the gut of living organisms. That's where we're going to find them, is if you use the washroom and you touch infected surfaces or what have you. Cook your food prior to proper temperature. Avoid drinking unpasteurized water. Practice good hygiene, such as avoiding contact, and get vaccinated against certain strains of E. coli. Now, you don't need to do any of those. A lot of these, like one of the things that really rings true to me is that E. coli is usually the culprit, or sometimes the culprit, in um, illness breakouts on cruise ships. And everybody gets all up in arms that cruise ships, they're dangerous, they're full of germs, blah blah blah, and they cause these outbreaks. But in real life, if you look at the number of these breakouts of the gastrointestinal problems are more common in group homes, such as nursing homes, the assisted care facilities, schools, anywhere that there's a bunch of food being made. But those are the primary two places that um, they find these outbreaks. And then cruise ships, I think, are number 3 or 4, which again has nothing to do with the fact that the cruise ship is necessarily dirty. It's just that one of the health precautions failed along the way. So there you have it. That's what I think we have here. I think we have E. coli. And again, this came from drills that we use every single day. Hello, this is Future Rich here. I forgot to touch on the third petri dish, which was the swab of the pen and the putty that I had been using to diamond paint with some of the very drills that I tested. Now, why did that petri dish not grow anything? I'm going to make just an assumption, a guess here, that perhaps there was not enough surface area when I swabbed the single placer end and the multiplacer end. And perhaps I just didn't pick up enough bacteria, or I didn't spin the swab to get that side of the swab onto the petri dish. I just can't fathom that the bacteria would mysteriously go away because of the pen or the putty or anything like that. So my assumption is that I just did something wrong in the experiment, and not that there wasn't any type of bacteria on the pen in actuality. Does this mean we should stop diamond painting? No. Does this mean that you're going to get sick? No. Does this mean that you should stop licking your pen? As I say in every video, there is no right or wrong way to diamond paint. You do you. And if you're a licker, continue to lick. Chances are you've already built up some type of immunity to this, or you haven't gotten drills that have had it on there, or maybe you have. Who knows? But just for your consideration. I hope you found this to be somewhat interesting, if not at least a little fun. I hope that we get to spend more time together in some future episodes of Speaking of Diamonds that perhaps could be a little bit less disgusting. Um, and just remember, like I always say, there's no wrong way to diamond paint. So when you diamond paint, you do you. Thank you and have a good rest of your day. Bye bye.